The first letter is for what? For the first name of the bacteria. Typically, bacteria, they have two names. The first name is the genus. The second name is what? The species.
genus and species.
Just like my first name, Kuteba, family name, Abad. So in bacteria, we name the bacteria using two names, the genus and the species. يرضى عليك باطلق على المحكمة. Now, the first capital letter is the first letter of the genus name. The second two small letters are the first two letters of the species name. So H from Haemophilus, I and D from what? Influent. Now, then we have what? We have some extra letters and maybe Roman numbers. These indicate that the extra letter, the D, indicates what? The strain of the Haemophilus influenza. There are different strains. Okay? No, okay. Escherichia coli, E from Escherichia from coli, R1 strain R, I, or 1, this is the first restriction enzyme discovered in what? In Escherichia coli. Number 3 means this is the third enzyme discovered in this bacteria. Okay? Branch 1, Bacillus ameliorations. We have the B, the AMI, H is the strain. I, or 1, this is the first restriction enzyme discovered in this bacteria. And as you can see, here we have what? The restriction site, AAGCT. Here we have another restriction site, GAATTC. The bandage one, GGATCC. This enzyme, and this enzyme. Okay. These three are called 6 cutters. 6 cutters means the restriction enzyme, the restriction site consists of 6 nucleotides. This one, the restriction site, consists of how many nucleotides? 1, 2, 3, 4. Okay? So it's a 4 cutter. Now notice, all these restriction enzymes, they generate what? Cohesive ends. They do not cut both strands at the same exact location. No. This enzyme cuts between the first A and the second A, here on this strand. On the other strand also, between the first and the second A here, generating what? Sticky ends, or cohesive ends. Same thing for equal 1, same thing for damage 1, and even 1, tag 1. But notice, tag 1, here we have what? You have to understand that this end, this is a sticky end or cohesive end. Okay? But here, what we call these is what? Overhang. Okay? Now, the overhang here consists of 4 nucleotides. The overhang here consists of how many nucleotides? 2. ما عاد حد يشوف فيهم 3 لأنه مش واضح البروجيكتور.
فيش يعني شايف خطوط تتبعي هون أحسن.
Why do you think this end is called the sticky end or cohesive end? Sticky means what? لازق cohesive. له علاقة بالمقابل للإيش؟ للانتصار. Okay?
عشان بقدر يرجع يعمل bind with.
The reason why these are called cohesive or sticky ends is that these ends could be brought back together if we use an enzyme called DNA ligase. So the idea here,
together.
when you have this double-stranded DNA fragment that contains this restriction enzyme called A. If you equal 1,
Ligase.
okay, the equal 1 enzyme
Ligase.
will cut this single strand, this one piece of DNA into two pieces or two fragments.
Ligase.
Now, these two fragments, now we have two separate DNA fragments. Each DNA fragment has blunt end, cohesive end, cohesive end, blunt end. We can take these two fragments and bring them back to their original state if we use what? An enzyme called DNA ligase. So we can go back from here to here using an enzyme called what? DNA ligase. Now, the DNA ligase, all what it does is that, as you can see here, we have what? The restriction enzyme cuts this phosphodiester bond and cuts this phosphodiester bond, which removes the A from the T, from the G from the A, and this G from the A. But this overhang that you see here is complementary to this overhang, which means they can what? They can base pair using what? Which type of bonds? Hydrogen bonds. So we have two hydrogen bonds, two hydrogen bonds, two hydrogen bonds, two hydrogen bonds. Okay? So basically the overhangs are complementary to each other. But the problem is that what? We still have what? This gap and this gap. There's no phosphodiester bond here. There's no phosphodiester bond. This is where the DNA ligase basically comes in, and the DNA ligase, what it will do, it will create what? A new phosphodiester bond here, a new phosphodiester bond here. Bringing the two stickings together, now we have what? We have another one piece of DNA.
To create.
Here we have also another example.
بين شو أوريها؟ بين مثلا هاي وهاي في بيناتهم دايسترسال اللي هي كيف بتصير هون؟
We have another one, HAE3 and SMAL1. Okay? These are the organisms from which these enzymes were discovered. And this is a 4-cutter, 6-cutter. But notice, these enzymes, they cut at the same location within the restriction enzymes at the two strands, generating what? Blunt ends, not cohesive ends.
restriction site كاملة.
Okay? Notice here, no overhang, no sticking. This is a blunt end, blunt end, blunt end, blunt end. Now,
إنه الأشياء اللي عادة المقطوم
طب البلانتس ايش؟
من البروجيكتور كثير سيء.
هاي البلانتس انت لسه بسأل عنها شو وضعها؟
لا الصفحة هاي كلياتهم حكى إنه إذا سكتوا the same side.
لما يكون السيم سايد.
Imagine if we have
طيب حكى عنها.
this is one DNA fragment that contains what this restriction enzyme for this enzyme. If we take this restriction, if we take this DNA fragment and this fragment is treated with what? Allo1. What will happen? Allo1 will cut this DNA fragment into two DNA fragments. Okay? So here we have fragment 1, fragment 2.
أعطينا فراغمنت.
Now, fragment 1 has two ends. This end is what? Blunt. This end, blunt. This end, blunt. This end, blunt. Okay? Here, the two fragments after they are the two fragments generated after cutting with this restriction enzyme, for example, this fragment has a blunt end, sticky end, blunt end. Okay. Now, what if we take fragment 1 and 2 and add to them what? DNA ligase. What will happen? How many DNA fragments will be generated? How many different DNA fragments will be generated? One.
لا لأنه بقولنا الفوسفودايستر بوند بلا.
Okay. What's the function of the
اسمها
DNA
لازم
ligase?
هيدروجين صح؟
فوسفودايستر بوند.
Make phosphodiester.
طيب
ماشي بس ما بعمل هيدروجين.
The two sticky ends.
ما بعمل هيدروجين.
You have to understand, these two sticky ends, they
هيدروجين.
come together because of hydrogen bonding between their complementary overhangs. All what the DNA ligase does, the DNA ligase will observe that there is a G next to an A, and there is no phosphodiester bond between them. The ligase will make this phosphodiester. Okay? The DNA ligase does not bring the two sticky ends together. What brings them is the complementarity between the overhangs. All what the DNA polymerase does, if the DNA polymerase observes or senses two nucleotides not having phosphodiester bond between them, it will join them together. If there is a free phosphatic group and a free OH. Okay? Now here, let me repeat the question. If we take these two DNA fragments, okay, put them in a distant tube, let's say, and then we add what? DNA ligase. What will happen? كيف يعني؟ Yes, the DNA ligase will ligate this DNA fragment with this DNA fragment to what? Regenerate this DNA fragment. What else? Yes. So here you have to understand. This DNA fragment, it's a 5' end, which means it will have what? Phosphate. And here, OH. What about here? Phosphate. Here, OH. Here, phosphate, OH. phosphatic group, and now the DNA ligase can join two nucleotides by phosphodiester bonds if one of them has a phosphatic group, the other has a free OH group. Okay? Which means it can join this phosphatic group, this OH group with this phosphatic group with
OH group.
generating this molecule. But imagine if you take one from this side to the other side, like here, what will happen? It can join this end with
This end.
this end. So it can give us what? Two different fragments. Here, what about here?
Just one fragment.
Is it possible that it will join this end with this end?
No.
Why not?
هو بس بدل أنا sticky ends على التراب.
Even if there is a sticky end. But the probability will be very low. Why? Because probably these two fragments, when they approach each other, the complementarity between these two overhangs will allow them to approach each other in this orientation. Here, no. These two fragments, they come closer to each other randomly. Sometimes one approaches two from this side and this side. In another instance, this fragment one will come from here, which allows the DNA ligase to what? To ligate this end with this, generating what? A totally new different.
Two different fragments.
Yes, both.
ممكن لما نحطهم بيتيو إنه هاي الجهة ترتبط مع هاي الجهة وممكن هاي الجهة ترتبط مع هاي.
So here you could say the results would be fragment 1 connected to fragment 2,
or 2 with 1.
okay, or 2
with 1.
connected to fragment 1. Okay. So this is the 3 prime, 3 prime, 3 prime, 5 prime, or the part prime would start from the 2 from fragment 2. Now, in DNA cloning, we prefer to use restriction enzymes that will produce what? Sticky. Why? We will discuss this later on when we start talking about cloning. The last type of, so in the previous lecture, we discussed polymerases, we discussed restriction enzymes, endonucleases, nucleases in general, DNA modifying enzymes, kinases, phosphatases, transferases, and the last type of enzymes that are used in recombinant DNA technology are the DNA ligases, which I already introduced to you. DNA ligases, their function is to make phosphodiester bonds. Here we have a double-stranded DNA. We have discontinuity in one of the strands. There is a missing phosphodiester bond the DNA ligase can make. Here we have two blunt-ended DNA fragments which will be joined together by the DNA ligase by making two phosphodiester bonds. But again, the DNA ligase, in order for the DNA ligase to make a phosphodiester bond, it will require what? Phosphatic group of one nucleotide and a OH group, 3' OH group from the other nucleotide. Now, DNA cloning, before we discuss DNA cloning, remember, scientists were able to do DNA cloning after the discovery of restriction enzymes and plasmid DNA. Now, plasmids are small circular pieces of DNA found where
Bacteria.
bacteria, but they are found in certain types of eukaryotic cells, like fungi. Okay, so we have eukaryotic cells, but they do have some plasmids. They are considered extra chromosomal DNA, which means they are not chromosomal DNA. But they are found in the same cytoplasm where you find the bacterial chromosome. What's unique about them is that they can replicate independently from the chromosome. So they can replicate themselves without having the chromosomal DNA of the bacteria replicate. Now, today we use plasmids as vectors. Vectors are viruses or DNA molecules that can accept, carry, and replicate whatever DNA you want. Okay? There's a whole section about vectors and their kinds and their uses.
متى بيصير البلازميد فيكتور؟ لما يحمل صفر جديدة.
Now, let's talk about creating a recombinant DNA. This is a topic that you've learned about briefly in Biology 102. Here we're going to remind you about DNA cloning and maybe expand further and see how DNA cloning could be done using different ways.
صفر مش صفر بدك تكتبها بطريقة بسيكونس أو
Now, DNA cloning began by using restriction enzymes and plasmids. Here, imagine, or here, what you see is human DNA, and we need to take a piece of this human DNA and put it inside what? Inside a plasmid. Okay. So the first step is to cut the human DNA and cut the plasmid DNA with the same restriction enzyme. Here in this example, it's equal. Why? Because the same restriction enzyme used to cut the human DNA that we want to clone into a plasmid DNA. The same restriction enzyme generates what? The same stick ends. So here, we have two cohesive ends or stick ends of the human DNA. This piece that you see in the middle, this is the one we want to clone into a plasmid. Now, the plasmid also must have the same cohesive end. So now we have two cohesive ends on the human DNA, two cohesive ends on the plasmid, and these two DNA fragments have compatible or complementary cohesive ends. So this is
هاي
compatible مع هاي.
this is what?
هم واحدة
This is the human DNA. This is the
بالفراغمنت
DNA
اللي احنا قلنا
fragment.
إياها واحدة بتكون هون
Let
بالبلازما.
me highlight it here. This is the DNA fragment that we want to clone. Okay?
نفس رسمتين أنا شغالة مش بطنها.
And now,
شو بي إيش؟
this is the same fragment I'm talking about.
ما ركزت كنتين.
But also, we have to cut the plasmid with the same restriction enzyme. Now, plasmid this is what? This is a circular plasmid. Imagine it has what? It has what? 1 restriction site. If we cut it with equal 1, what will happen?
أخي نفسي زبطت
We will have
نحكي بأرقام
sticky.
ما نحفظ اللي يضبطها. مش شايفة.
سيركلر بلازما. لا هاي كلها سيركلر بلازما.
So this is one sticky end, this is another sticky end. Okay?
مش هذا. ارجع انزل ارجع.
Now this is
الله.
a linear plasma. It's no longer a circular plasma. But now this linear plasmid has sticky ends that are complementary to the sticky ends on the DNA fragment that we want to clone. Okay? So the next step, after having a linear plasmid with these two sticky ends and the DNA fragment with these two sticky ends, we put them together, okay? And we add what? DNA ligase. Now, before you add the DNA ligase, this DNA fragment and this DNA fragment, the DNA fragment we want to clone and the linear plasmid with these two sticky ends, they can come together like this. Okay? Why? Because of hydrogen bond.
مع بعض عشان
But
come together because
here we have what? A gap, a gap, a second gap. So we have 4 locations within this circular plasmid that are missing phosphodiester bonds. If we add the DNA ligase what? Will seal these gaps, will make phosphodiester bonds, causing the formation of what? A stable plasmid. This is unstable, which means at any point, the two fragments, this fragment could go out of the plasmid. But if we add the DNA ligase, now we have what? A circular plasmid. Okay? This circular plasmid is carrying what? It's carrying the human DNA. I will highlight the human DNA.
اسمع مزن والله انني
Okay?
استبقى الأحلام.
قبل لا كنت مستبقة بس انت هاي قبله. شوفي.
What's the difference between this circular plasmid used before the cloning and this circular plasmid generated after تصوريش؟ يلا مباركة. ممنوع التصوير.
nucleotide.
ممنوع التصوير. And this plasmid generated after the clone. This is a circular plasmid. This is a circular plasmid. What's the difference between them?
Modifications.
Yes, here we have one. No human DNA. Human DNA. What else?
ما سمعتش.
إنه الهيومن DNA fragments اللي
Okay, because here we have non-recombinant plasmid. This is what? Recombinant DNA. Why? Recombinant because it has 2 DNA from 2 different sources. The plasmid, this DNA highlighted with red, is human DNA. The rest of the plasmid is what? Bacteria DNA. What else? الكل مرة نسجل basic biotechnology. وين يا شيخ؟ وين باقي؟ قربنا نخلص.
5.
هيك بدك تقضيها الفصل؟ نعم. ليش بتخلص؟
زين.
مش سامع.
أزمة.
أزمة. This means ممكن هذا دي ان يو جي. برافو عليك. Here we have what?
إيكو أروان.
One restriction site for what?
إيكو أروان.
Here we have what?
شو؟
Two.
والله حاسة. هذا قطع.
Where does these? Why we have two here? The plasmid
One.
had only one, and now it has two restriction sites.
نفس الريدة
Because
instruction site.
we have 1, 2, 3, 4 stick ends, which generates, they come together and generates two restriction sites. Okay?
سؤال جواب عليه. شو الفرق بين هذا وهذا؟
So this is one
هاي فيها
restriction
two
site,
restriction site وهاي فيها بس واحد. ليه؟ مو عدلنا عليه واحد.
and
بس
this is
أنا
تفهميني يا بعدين لأنه إحنا ما نفهم.
and both restriction sites
بستر عليه.
can be recognized by the enzyme EcoR.
حطت لي كرنيل في رياضة زيرو بهتان.
هو المكتوب أنا مش شايفة. حسن كنت خليت دخلنا في آخر شيء مزبطوش ولا إشي.
Okay, this is the general procedure used for DNA cloning. This is the procedure used in the early attempts to clone or to produce recombinant DNA. Now, this is recombinant DNA. You can call it also recombinant plasmid. Okay. What do we do with this recombinant plasmid? The next step is to take this plasmid, put it back where? Inside bacteria. Okay? You have to understand that we get the plasmid from the bacteria,
أو injection.
we use it to make a recombinant plasmid, then we take the recombinant plasmid back into the bacteria. Why? Yes, because we need to make millions of copies of this plasmid, which means we will be making what? Millions of copies of the DNA we clone. And this is the purpose of cloning. Okay. Now, we can take the plasmid, we put it inside bacteria. What will happen inside the bacteria, the plasmid will be replicated, so the bacteria will have multiple copies of your recombinant plasmid.
البكتيريا بنمسح
And then
البكتيريا
the bacteria will grow inside, will start dividing, increasing their number. So you will have what we call
بيجي
colony.
بعمل ريبليكيت.
Okay. So we put the plasmid inside the bacteria. Then we allow the bacteria to grow on the surface of a growth medium or plate. They will form a colony. Then we take this colony
بكتيريا.
and we allow the bacteria to further grow in a liquid type of food. So now you will have billions of cells. Each cell will contain dozens of your plasmid. Okay? Then you can take the plasmids out of the bacteria and do whatever you want to do.
ما حكى طريقة إنه نحط
Now,
البكتيريا
sometimes
البلازما
we
جوا
do cloning.
البكتيريا.
أتوقع نفس
أنا بعرفها بس ما حكى مثل النوع
اللي نفس هاي اللي هون.
So why we do cloning? What's the final purpose of cloning?
هاي فيروس
Why do we
والبكتيريا.
clone
هاي إحنا عدلنا من رجع البكتيريا طلعنا رجعنا.
DNA fragments, for example, from the human genome into a bacterial plasmid? What would be the purpose of cloning a DNA fragment into a bacterial plasmid? produce what? A protein, right? You can clone a gene and express the gene to produce a protein. What else? ليش نقول إصلاح؟ study the DNA, okay?
إذا ما حكيتي كيف
Study, let's say the gene
هدول
is
كانت
an
بالشابتر الأول.
أو DNA,
هدول.
okay?
هون زي الأنسولين.
ذا جين
All the DNA.
جين
الجين
DNA or DNA.
جين ولا أور؟
جين
What else?
ذا جين أو دي إن إيه حطي جين شوف
Sometimes we want to do what?
استني.
Sequencing the DNA. We want to determine the nucleotide
طب ما هي
sequence. So sometimes we have to clone it into a plasmid. Okay?
sequencing.
زي كمان أنا برتب
Yes. Maybe we want to do cloning. زي ما حكى زميلكم يوسف إنه إيش؟ Change the DNA sequence. We have a DNA fragment or a gene that we want to change its sequence. These are the four major reasons why we need to clone, but there are many others.
فكرة أسئلة كيف دخلنا
But following the generation of the recombinant DNA, we have to do two things.
شو إيش؟
The first thing is what? Transformation, which is what? Adding the plasmid to bacteria. Then we have to do what? Selection. We have to select the bacteria that will have my plasmid.
لا لا شوف
أول إشي ندخلها جوا البكتيريا بعدين selection.
So, getting new recombinant DNA inside bacteria is called what?
transformation.
Transformation. Okay? Transformation could be done into bacteria
هاي بحصل
by two different ways. The first one
injection or insertion.
is considered what?
chemical methods.
The chemical method. In the chemical method, we start with what? With a normal bacteria. Now, this is the bacteria that you want to accept or take your recombinant plasmid. Now, before this bacteria can take your plasmid, it must become a competent cell. Competent cell, this is a bacteria ready to take any foreign DNA, aka your plasmid. So we treat the normal bacteria with
شو
what?
إيش؟ جديد شو
many DNA.
With a chemical called what? Calcium chloride. Okay? Sometimes we use another chemical called rubidium chloride. Both are chloride salts. Okay? This one.
تكتبها.
بعلق يعني
Now, most of the time, the bacteria we use for cloning is what? Is the E. coli. Now, if you treat the E. coli cell with rubidium chloride or calcium chloride, it will become what? Competent cell. Now, you take your plasmid, your recombinant plasmid, this plasmid, you mix it with what? With this competent bacteria in one test tube, and then what will happen, the plasmid will stick to the cell wall or to the outside of the bacteria like this. So this is the plasmid. Now the plasmid is still outside the bacteria. To force the bacteria to take the plasmid, you have to
exterior.
subject the bacteria into a heat shock. Okay, at 42
هيك نكتب شو؟
Celsius. So basically,
شو؟
you take your plasmid, you take this bacteria, you mix them together in one test tube, you give them 5 minutes,
شوك من الشوط.
so all the plasmids will stick on the surface of the bacteria,
صوت
and then you heat shock
شوك.
the bacteria. You increase the temperature of the bacteria at 42.
شوك نظبطها.
For only a short period of time, 1 to 2 minutes, no longer than that. Because 42 is not an optimum temperature for the growth of the bacteria. It can kill the bacteria. But the 42, this high temperature, what it will do for the bacteria, it will open some transient pores on the surface of the bacteria, allowing the plasmid to go inside. Okay? Then you have immediately to take them after the 2 minutes at 42. Typically we do it from 1 minute. After 1 or 2 minutes at 42, you have to take the bacteria, quickly put it on ice. You have to cool it down very quickly. Otherwise, the bacteria will be killed. So this heat shock
Nice.
opens an opportunity for the plasmid to go inside the bacteria. Then you take this bacteria that has the plasmid, put them on ice so that the temperature of the bacteria drops significantly and now the bacteria stay alive. And then you take this bacteria and put it inside where on the top of a medium plate to allow it to grow, to allow it to divide, and to allow the bacteria to replicate your plasmid. Okay? So this step
injection theory.
is called what?
اوكي. نسيتها هذيك الطريقة.
Another way
هيها. شوي. هيها.
of introducing your recombinant DNA into bacteria
Hold on.
is by what?
Electroporation.
Electroporation. Okay? We have electro from
بس برضه نصيفة.
electro means what? Electric. Electricity. كهرباء. Poration.
علي.
Support.
بتعلمش.
Okay? So here, instead of using heat to make pores on the surface of the bacteria, here we use what? Electricity. Okay? Now, again, you take your plasmid, you mix it with bacteria, okay? And then you apply a brief pulse of high voltage electricity. This is a high voltage electricity. This high voltage can kill a human being. But this high voltage is only done for microseconds or milliseconds, for a very brief short of time, because this high voltage can also kill the bacteria. But because it's done very briefly, it cannot kill the bacteria, but it can do what? Create tiny holes or pores through which the plasmid will go inside the bacteria. Okay. So here,
إنه الكهرباء عشان تعمل خطوط صغيرة في السلوات. تفكري لما احنا تمسح شو من هون.
we have a device called the electroporator, which can generate
بس هو أنا بس أحكي إنه بس شيل الشمس.
high voltage, 10,000 volts, for what?
شو
10 milliseconds. 10 أجزاء من الثانية. So you put the cells
شو ربطش
and the DNA in
من
one tube, then you put the tube inside the device, and the device will apply this high voltage or electric pulse. Okay. So we have two types of transformation methods. We have chemical method, then we have what? Electroporation. These are the two major methods used to introduce plasmids into bacteria. But there are other methods that could be done to introduce DNA into other types of cells. So for example, into animal cells, we can do what we call micro-injection. Using microscopic needles,
حلو حد رح حفظتها
you
بالضبط.
can put the DNA inside the needle and inject the needle directly under the microscope into what? To the cell nucleus. Because eukaryotic cells, human cells or animal cells, they are much bigger than bacteria. We can see them under the microscope, and we have microscopic tools that could be used to produce DNA inside the nucleus. Or we can grow cells, animal cells or human cells, as a monolayer, as one layer, and then we add calcium phosphate to them, okay, on their surface. Then we add what? The plasmid. Now the cells will take the plasmid by what? By endocytosis. And then the plasmid will go into the nucleus. Or we can do something called what? Liposuction. Liposuction means we put the DNA inside what? Synthetic, let's say, liposomes. Liposomes are bodies that only consist of what? It's a ball of phospholipids or lipids. Okay? So these liposomes, okay, they have only what? A membrane consists of lipids, and inside this membrane we put what? The DNA. Now the phospholipids in the liposomes, they are similar to the phospholipids found in the membrane of the cell, which means if the liposome reaches the cell, its membrane will fuse with the membrane of the cell, releasing what? Releasing the plasmid or the DNA into the inside of the cell. And from there it can go to the nucleus. Okay?
the DNA ولا هذا
بس ام دي ان الله اكبر الذكاء.
خلص مش عارفة نرجع نطلع.
هذول الليبسومز هم نفسهم تقريبا
نحل الثاني
يستخدمون حتى يوصلوا الأمعرنة فاكسين تبع فايزر لوين
سوريا
انت اخذتي فايزر؟
الفايزر
هاي فايزر
شو هي؟
تبع كورونا.
خلاص يا رب.
دكتور كيف ببغى احضر احنا بغلي من القهوة لازم اكل شيء.
نستنى
شو ايش اذا بدك شيء انه
اللي يستخدمون